Well, thanks very much, Sarah, for the kind introduction and also for the invitation to present this webinar. I'm really delighted to be here and to share some of our latest work with the listeners. So the work that I'm going to tell you about today has to do with our interest in developing next-gen therapeutics targeting HIV integration. And this particular biological process, the integration of the viral genome into target host cells, has been of interest to the lab for some time now, and we've described some of the structural biology of the nucleoprotein assemblies that perform this process. But this process is also targeted by some of the latest and really the best available antiretroviral therapeutics, and there's active chemical development in the area. So with some of the continuous developments in cryo-EM, more recently what we realized is that we can now utilize advanced imaging and image processing strategies to understand how small molecules bind to these nucleoprotein assemblies that catalyze integration, and really to utilize the workflows for structure-based drug design. So I'm extremely excited to share this story with you today. So my lab is a structural biology lab. We use cryo-electron microscopy, or cryo-EM, to understand macromolecular structure. Why study structure? Well, structure describes function. So we are interested in not only understanding how macromolecular complexes are assembled, but why certain types of assemblies might occur. We're interested in the atomic level details at the level of individual residues and base pairs, which of these may play functional roles. We want to know how to perturb macromolecular function by modifying the structure, and ultimately would like to harness the information to improve human health, so thinking more specifically about rational drug and vaccine design. And finally, we're also interested in methodology development to facilitate, not only to facilitate structural studies, but also to open up novel areas of investigation. So the main structural biology technique that we employ is single particle cryo-EM. Now the way it works is briefly shown on this slide. First, one needs to purify a sample of your macromolecular object or macromolecular assembly of interest. You then apply that sample onto a 3 mm cryo-EM grid. The grid is plunge frozen into liquid ethane, and the sample is vitrified or frozen in its native hydrated state. This frozen sample is then inserted into an electron microscope, and you start to acquire images. Now these are the types of images that one can obtain, and what you see are individual monodisperse single images of, or single particles of, in this particular case, the GRO-EL macromolecular assembly. So we refer to these as individual particles. We can select them out and then subject them to a number of image processing operations that will ultimately allow you to arrive at a three-dimensional reconstruction. And if the reconstruction is resolved to a sufficiently high resolution, then one can interpret it with an atomic model. So cryo-EM is by no means a new technique. It's been around since the 1980s, and really some of the early pioneering work was conducted by Bob Glazer and Ken Taylor in the 1970s. And there are a number of important developments in the field, and these are highlighted here. First of all, the vitrification technology. Vitrification was pioneered by Jacques Dubochet throughout the 80s. It allows the experimentalist to freeze macromolecular specimens in their near native or frozen hydrated states, so the sample doesn't get a chance to crystallize. And vitrification of macromolecular specimens really forms the basis of cryo-EM. A number of early image processing procedures were developed also in the 80s for example, assigning orientations to individual particles, for performing reconstructions, and for many other steps of the cryo-EM workflow. Computational methods really continue to constitute the backbone of current development. And there were also important demonstrations of what cryo-EM can do. In 1991, the first atomic structure of bacteria rhodopsin was determined using a type of cryo-EM called electron crystallography, cryo-electron crystallography. And this showed that we could really gather important structural information about macromolecular assemblies. So for these and other developments, these three individuals, Jacques Dubochet, Joachim Frank, and Richard Henderson, were awarded the 2017 Nobel Prize in Chemistry for their work in developing cryo-EM for the high-resolution structure determination of biomolecules in solution. Now, since those early developments, there have been a number of important advances in more recent years. First of all, direct detectors came on the scene about a decade ago. Now, these detectors first allowed us to acquire images with increased signal-to-noise ratio, both in comparison to conventional CCD detectors, but also in comparison to photographic film. And second, they allowed us to record movies instead of still images. Now, the reason these movies are important is because the specimen is actually not completely still under imaging conditions. So two distinct phenomena, beam-induced movement and stage drift, lead to blurry images. However, if one records movies, then you can align the individual frames and you can account for this movement, which then leads to crisper images. Algorithm development has played a huge role. Software such as Reline, CryoSpark, System, they've really revolutionized how much we can extract out of noisy cryo-EM images. And finally, automation also played an important role. Software such as Leginon and cryo-EM have allowed us to acquire really huge data sets. And this is necessary not only for pushing resolution, but for making sense of increasingly more challenging and heterogeneous samples. Now, all these developments led to what we now refer to as the resolution revolution. Basically, cumulative advances in the field have allowed us to solve very important macromolecular complexes. The ones that are displayed on this slide here are the mitochondrial ribosome from Venke's group, the trip channel from Yifan's group, and the F420 reducing nickel iron hydrogenase enzyme from Werner Kuhlbrand's group. And this revolution is really allowing traditional microscopists to become true structural biologists. So we've seen steady improvements in resolution since that time. That was around 2013, 2014. In the last several years, a number of sub-2 angstrom resolution structures have come out. So in the first panel A, it's the beta-galactosidase enzyme from Sriram Subramaniam's group. In B, that's the adeno-associated virus from our group. And C and D, there's several apoferritin structures from the groups of Radodanev and Shor-Shares. Now, the ones on this slide are actually from early 2019. Since then, we've seen a number of other structures come out in the 1.5 to 2 angstrom range. And I would say we can fully expect these advances to continue for years to come. So we're still not at the limit, at the optical limit of the microscope. So of course, people started to take notice. This is a slide showing the cumulative number of map depositions into the electron microscopy databank. You can see that around 2013, there seems to be an inflection point, and we're now at a time where the technique is increasingly growing in popularity. So this increasing popularity also translates into atomic model depositions. This is a recent tweet from Masahiro Kikawa showing the cumulative number of model depositions into the protein databank, and specifically in comparison to other methods, namely X-ray crystallography and NMR spectroscopy. So we are now in 2020. Okay, we're still about a factor of 5 behind X-ray crystallography in terms of the number of released structures per year in the PDB. But if you extrapolate this line over the last 10 years and extrapolate it forward, we really seem to be on an exponential growth curve. And so this means that by around 2023, 2024, we'll be approximately neck and neck with the crystallographic community in terms of the model depositions. So that brief introduction, I'm going to get into the meat of my talk. Building on these developments that I've just introduced in cryo-EM, what we really wanted to do is to ask whether we can now utilize these increasingly powerful techniques for the purposes of drug design targeting HIV integration. Now, my lab is interested in HIV structural biology. We've worked in this field for almost a decade now. We continue to work in this field in part because of this slide. If you look at the prevalence of global HIV infection in first world countries, the infectivity rates are relatively low, ranging typically less than 1%. However, if you look at other countries, such as Russia, such as Thailand, and virtually every single country in sub-Saharan Africa, these rates are significantly higher. So as of 2017, this is data from the CDC, there are about 37 million people who are infected with HIV. There's about 2 million new cases per year and about 1 million deaths, which means that we are now in 2020, so there's about 40 million people who are infected or living with the virus. Only about half, at this point, a little bit more than half, are on antiretroviral therapy. Okay, so we need to do something to tackle this really global problem. So let's start with a little bit about the viral life cycle. So these are the early events that are displayed on this slide. The virus is at the top. It's an enveloped retrovirus, which means that it contains a lipid bilayer that is of host cell origin. On the surface of this bilayer, there are proteins called enveloped glycoproteins. Now, these proteins bind CD4 receptor and either the CCR5 or CXCR4 co-receptors, which then triggers a conformational rearrangement and leads to the fusion of viral and cellular membrane. Fusion then releases the conical capsid core, which contains 2 copies of genomic RNA. This core is trafficked along microtubules to the nuclear pore complex, and during that time, the genomic RNA is converted into complementary viral cDNA through reverse transcription. Reverse transcription, that's really one of the hallmarks of all retroviruses. After completion of reverse transcription and synthesis of viral cDNA, a multimer of integrase proteins can then bind to the ends of viral DNA to form what's known as a pre-integration complex, or a PIC. And at some point during that whole process, from the conversion from the reverse transcription complex to a pre-integration complex, there's also an uncoating step and a translocation step to the nuclear pore complex. The timing of all these events is still poorly understood. But once in the nucleus, the PIC can scan target chromatin and identify a select region for integration. Integration then establishes a permanent and irreversible infection in that target cell. So we are specifically interested in the steps involving the PIC. The PIC is a large and still enigmatic assembly. There's 10,000 base pairs of viral DNA. There's integrase, multimers of integrase. There are cellular host factors, probably some capsid, a viral capsid, and probably unknown other factors that are involved. It's a big, messy complex, and it hasn't been really amenable to homogeneous isolation for structural studies. But what we can do is we can assemble complexes that contain the basic functional components of a PIC in vitro. So these complexes are called intasomes, and intasomes provide an in vitro system for studying the cellular PIC. What we do is we have oligonucleotides of viral DNA. So these are typically 25 to 27 base pairs in length, and we incubate them with integrase proteins. And under specific biochemical conditions, those can then form into a complex, into a nucleoprotein assembly that we call the stable synaptic complex. Integrase will then cleave off 2 of the nucleotides from the 3' end to form the cleaved synaptic complex. That complex can then capture additional target DNA, so additional oligonucleotides to form the target capture complex. And finally, integrase will perform its second catalytic step to make 2 nicks in target DNA and insert viral DNA into the target DNA. So that forms a strand transfer complex intasome, or STC intasome. A number of years ago, we described the structure of the HIV strand transfer complex intasome. It's shown here. The lentiviral intasome is a large nucleoprotein assembly composed of viral integrase and nucleic acid. In the STC intasome, we see 2 DNAs. First, there's the viral DNA. That's in red. There are 2 copies of viral DNA. These represent the 2 ends of the viral genome. So in reality, these ends are slightly distinct. There's a U3 and a U5 end, but in our case, we're simply using 2 identical oligos for the U5 ends. There's also the host DNA, and that's shown in orange. Now, this particular complex represents the product of strand transfer, and therefore both the viral DNA and the target host DNA are actually joined together as a single molecule. And the DNA, in turn, is bound by multimers of viral integrase proteins. So in this assembly, there are 12 integrase proteins that form an oligomeric assembly around the nucleic acid. There's actually a range in terms of the number of integrase enzymes that can assemble around the DNA ends, but a few of these really have to be conserved in order to form a functional intasome. So there are a number of strand transfer inhibitors that have been developed over the last actually several decades. And 4 of these have been clinically approved by the FDA. Raltegravir and elvitegravir, these are first-generation inhibitors, whereas dolutegravir and bictegravir, these are second-generation inhibitors. Bictegravir is the latest to have been approved by the FDA. It's been approved in 2018. What's important is that all of these compounds, they bind intasomes. They do not bind free integrase. So intasomes are the primary targets for these leading clinical therapeutics. As of today, there are approximately 40 or so drug-resistant mutants, and counting, they keep increasing. So we really want to understand not only how these compounds bind to the HIV intasome, but also how the virus acquires resistance. So getting back to the assembly mechanism that I introduced a couple of slides ago, what we know about these inhibitors is that they work at this particular step, so they inhibit the formation of the target capture complex. And this also means that they bind to the cleaved synaptic complex. So we can assemble the cleaved synaptic complex bound to these inhibitors, and we can utilize, and then we can study these assemblies using structural biology. So we can purify these assemblies. Just to briefly mention the purification scheme, we actually use 2 gel filtration steps. The first step uses a conventional Super 06 column. And even though we get a single peak on the S6, actually the mixture is still quite heterogeneous. And we found this out after we started to use these aggregate columns, TSK gel aggregate columns. So if you take a purified peak from an S6 column and inject it onto the TSK gel column, you can see that there's still quite a heterogeneous mixture, and that's represented by the blue and the red profiles. I'm not going to get into what these CSC stacks are. I'll just say that we can take the peak that represents the CSC intasomes and then use it for structural biology. And we know that it's at least reasonably clean, because if we re-inject it onto the same column, there's largely 1 peak that remains, although the purification is by no means perfect. And then, if we take the intasomes and subject them to an integration assay, we see that they are competent for cleavage, and that's shown in lane 2 of this gel, where you see a smear that represents multiple cleavage events. But if we use inhibitors in lanes 3 through 6, integration is effectively blocked. So this is what the cryo-EM data looks like. Here you see a raw micrograph and 2D class averages from the data. You can see that there's a little bit of aggregation that's going on here. This has to do with the fact that the particles adhere to the air-water interface. It's by no means an ideal distribution. This really is a sort of a typical micrograph, but nonetheless we can recover some nice signal from these particles and get reasonably looking class averages. Now, our very first attempt to resolve the structure of an INSTI-bound intasome resulted in a map that looks like this. This is a view of the active site. This is where INSTIs bind. And you can see that there's a significant amount of smearing that is occurring. This has to do with the preferred orientation problem. Okay, we have one and only one preferred orientation of these particles in the data. We know from Alex Noble's work that preferred orientation is really one of the most common problems, current problems in the cryo-EM field, at least using current vitrification strategies. And what we suggested a number of years ago is that you can tilt the grid in the microscope to help to alleviate resolution anisotropy and this smearing that's caused by preferred specimen orientation. So the idea is that if you have a cryo-EM grid, and so here's a cross-section of the grid, particles tend to absorb either to the top or the bottom, so one of two air-water interfaces. And when you collect data for samples that exhibit this sort of behavior, you really only get one view of your macromolecular object of interest. However, you can tilt the grid during data collection inside the microscope, and when you do that, you can get a much better set of orientation distributions and more comprehensive sets of views. So when we did this with HIV intasomes, we found that in the absence of tilts here on the left, you get a very anisotropic reconstruction. However, if you just tilt the data, in our case we used 20, 30, and 40 degrees, we can get a much better and a much more isotropic reconstruction. So just briefly as an aside, there are actually a number of benefits to tilting for these preferentially oriented samples. You can improve the homogeneity of the orientation distribution, so these are your Euler angles. You improve the directional resolution by virtue of a more homogeneous Euler angle orientation distribution, Euler angle coverage, so you get less anisotropy. And then in a more recent manuscript, we actually showed that you can improve the global resolution by virtue of improving a factor that we've recently introduced called the sampling compensation factor. So these are the maps that we obtain using the workflow. On this slide, the same map is shown in 3 different ways. On the left is the raw map. In the middle is the same map colored by the Integrase protomers surrounding and bound to the lentiviral DNA. There's 8 of them, or at least domains of 8 Integrase protomers. And on the right is what is known as the conserved intasome core. Now, the conserved intasome core is comprised of Integrase protomers that are, as the name implies, conserved among retroviral species. So not all intasomes are so large. Not all of them form hexadecamers or dodecamers. Some are octameric, still others are tetrameric, but there's always the conservation of these central domains that are colored in yellow. And these conserved domains are the ones that perform the catalytic process. So getting back to the map on the left, you can see that there's a lot of heterogeneity in the peripheral regions. This again has to do with the incomplete assembly. Okay, but what's important is that the regions that are outlined here in red, that's the catalytic site. Okay, so that's where INSTIs bind. These are also the best resolved regions in the map. Now, we used 4 different INSTIs for our structural studies, and these INSTIs are shown here. Bictegravir, this is the leading clinically approved drug. This was approved by the FDA in 2018. It's one of the best of many drugs that are used to treat HIV-infected individuals. And these 3 compounds, these are developmental compounds. These are based on a distinct naphthyridine scaffold. They've been developed by our collaborators at the NIH, Terry Burke, and in collaboration with the virology lab of Steve Hughes. So these compounds are based on a different scaffold. And some of these compounds, in particularly 4D and 4C, they are extremely potent, and in fact more potent than bictegravir when used against viral resistant variants. Now, for those who are interested, this is our table 1. Just a few things to highlight here. We used the Krios with a K2 detector, operating in counting mode. We actually used a range of tilt angles from 20 to 40 degrees. Now, in principle, you only need 1, but we didn't know which one would work best, so we used a small range. We collected with a dose rate that would minimize coincidence loss on the K2, while at the same time maximizing the information content within each frame. So that was about 5 electrons per pixel per second. About 2,000 micrographs per data set, which after cleaning resulted in about 150,000 particles within each of the final maps. And these maps were on average resolved to 2.6 to 2.7 angstroms within the active site, which is where INSTIs bind. The one exception was compound 4C, which was slightly lower resolution. So, as with any other sample, we used a fairly comprehensive strategy for cleaning the data. We used Motion Core 2 for movie frame alignment, warp for CTF estimation and for particle picking. And then we moved directly to 3D classification and refinement. We actually deliberately skipped the 2D step. We found that 2D classification tended to weed out good particles that actually had a positive contribution to the resolution of our final map, so we skipped that step. 3D classification was performed iteratively. So we separated the data into two classes. We then discarded the particles from the bad class, kept the particles in the good class, and then repeated this iteratively until we saw no more further improvements. And finally, we performed beam tilt refinement, particle polishing, and this led to the reconstructions that were, on average, resolved to about 2.8, 2.9 angstroms globally, and about 2.6 to 2.7 angstroms within the catalytic core. This is just showing the resolution analysis. On the left, the conserved intasome core is colored by local resolution. Again, the core is resolved to about 2.6 angstrom. The peripheral regions are in the 3 angstrom range or below. On the right, you have the global FSC showing a global resolution of about 2.8 angstroms. And below that is the Euler distribution plot and a central slice through the 3D FSC. The slice is shown for the cross-section for the most anisotropic view, which is actually not bad considering that we started with highly anisotropic reconstructions. Now, all the analyses that I'll describe from here on out were performed with the maps that I just introduced in the previous slide. So these maps resolved to about 2.6, 2.7 angstroms in the active site. But actually, in the last several months, we saw this tweet from Tom Terwilliger showing the new density modification tool implemented in Phoenix, which improves the phases of cryo-EM maps. So he suggests that one could see, on average, 0.2 angstrom improvements in density maps for the cases that were evaluated. And it turned out that this was also approximately what we saw when we applied the approach to our intasome reconstruction, so also about 0.2 angstrom improvements. So even though the analyses that I'll describe were performed with the low resolution maps, we are now really quite excited that we have higher resolution data and with which to evaluate the reconstructions, and more generally about this density modification approach, which seems to really be helpful for many different data sets. So INSTIs bind to the conserved intasome core. These are, so there are 2 active sites, and accordingly 2 regions where an INSTI would bind. This slide just shows the conserved intasome core for the map bound to the compound 4D. In yellow, that's the density of the 4D compound, and on the right is the atomic model of the ligand. Here, we are now looking into the density of the active site for all the 5 reconstructions that we analyzed in the course of this project. So that's the APO reconstruction and the reconstructions with 4 compounds, bictegravir, 4F, 4D, and 4C. So in all cases, the ligand is completely resolved. Another thing that's worth noting is that we have many water molecules that are surrounding the ligand. And that particular aspect will become more obvious in the next slide. So this is a movie of the density in the atomic model of the reconstruction containing the compound 4D bound to the active site. You can clearly and completely see the density of the entire drug. We can refine it into the map without really much ambiguity. The compound binds 2 magnesium ions. So both of these magnesium ions are resolved in the maps. You can see the 2 large bumps that are chelated by the bound ligand. Magnesium ions are actually critical to the enzyme. They're involved in the catalytic mechanism of integrase for the cleavage reaction. So what these compounds do, what these INSTIs do, is that they chelate the 2 magnesium ions, and therefore they block the enzymatic cleavage reaction. Surrounding the ligand, you can see that there are lots of water molecules. There's about 10 of them that are evident in this movie, and more that are surrounding the active site. So these water molecules are critical. They're relevant to drug binding. They can be exploited, as I'll show in a later slide, for the purposes of drug design. And one of the things that we came to appreciate throughout the course of this project is the relevance of bound water molecules. So you really only see them once you start getting to sub-3 angstrom resolution. But in this particular instance, that extra fraction of an angstrom was really important for improving our understanding of the active site and the ligand binding. Here we show more details of the binding mode. This is bictegravir bound to the active site. You have a central scaffold containing electronegative atoms. These electronegative atoms, they chelate to magnesium ions. This is common to all INSTIs. On the right, you have a halogenated benzyl ring. This ring forms pi-pi stacking interactions with, or a pi-pi stacking interaction with one of the bases of viral DNA. Now, this specific interaction between the halogenated benzyl moiety and a viral DNA nucleotide, this is precisely the reason why INSTIs bind intasomes, but not free integrase. It's the reason why we need to assemble these complexes, these nucleoprotein complexes, in order to understand their binding modes. Here we show how the binding mode of bictegravir compares to that of the compound 4D. Now, as I mentioned earlier, 4D is a developmental compound. It's synthesized by our NIH collaborators, the group of Terry Burke, in collaboration with the group of Steve Hughes. 4D is a leading preclinical candidate, and in fact it has higher potency than bictegravir against many drug-resistant variants. So with 4D, we can see that the core scaffold is shifted closer to the magnesium ions. And that's because it's based on distinct chemistry, where the electronegative atoms are actually part of the central naphthyridine core. We also see this long extension on the left side of the molecule. This extension is very important. It contributes quite significantly to viral potency. It also constitutes the region that we are actively exploring and modifying in order to try and identify compounds with higher potency against viral resistant variants. Now, I already mentioned water molecules. We see them throughout the active site. This slide shows the density and the location for some of the bound water molecules in the active site of the apoform. The apoform contains calcium, as well as 3 of the INSTI bound forms. These INSTI bound forms contain magnesium in the active site. Medicinal chemists like to look at figures like this, which simplify the analysis. This is a 2-dimensional schematic of how the INSTI binds to the intasome active site. INSTI interactions and waters can both be subdivided by their relative position with respect to the plane that is formed by the magnesium coordinating the ligand scaffold. So there's the core naphthyridine scaffold. This chelates the magnesium ions and also contains the 2 R groups. R2 contains the halogenated benzyl moiety, stacks against viral DNA. The R1 group, that's the one that we're very interested in, and the group that we're actively modifying to elicit better binding and better response. It points toward the bulk solvent. What's important is that the positioning of its long chain displaces loosely bound waters. Now, I'm not showing the data here, but in addition to the structural interpretations, we actually have some MD data that confirms the water molecules in the bottom left region of the schematic are loosely bound. And displacement of these loosely bound solvent molecules should be entropically advantageous for the compounds. There's a large network of water molecules above the core scaffold and also below the core scaffold, and we believe that both of these networks can potentially be exploited for the purposes of drug design. So this is where future computational chemistry efforts will be important in order to really understand the energetics of binding and the favorability of water displacement. And we're actively investigating this area in collaboration with Stefano Forli at the Scripps Research Institute. So I do want to note that while the HIV structures have certainly been very informative, we did know quite a bit about INSTI binding through earlier work. About a decade ago, the Cherepanov group solved structures of prototype foamy-virus intasomes. These are intasomes from a simpler retrovirus, and the complexes were also assembled and solved with several bound INSTIs. We learned a lot from that early work, a lot about INSTI binding, and also about mechanisms of viral resistance. The problem is that the active sites of HIV and prototype foamy-virus intasomes are not conserved. Depending on how you align, the conservation of the catalytic core domain is around 20 or 30%. And there's only about a 40% conservation in the residues that are mutated by the virus when it evolves resistance against INSTI-based therapy. This is really important. It's important because when you overlay the two active sites, they're not identical. And this figure shows the RMSD between the prototype foamy-virus intasome active site and the HIV intasome active site, and it's mapped onto the structure of the latter. What one sees is that while much of the active site is similar, there's a substantial part that is not. There's a substantial deviation, especially in the part in the bottom part of the enzyme. The residues that are highlighted in this figure, these are residues that are frequently mutated by the virus in patients. These are viral resistant variants. Many of them are not conserved between the two retroviral species. The consequences of this lack of conservation are profound. If we take one particular compound, the compound 4F, this is a compound that has a sulfhydrobenzene extension on the left side of the active site as its R1 substituent, and we look at how it binds to the HIV intasome, we see that the chemical group extends out away from the active site. You can see that on the left. However, that exact same compound binds very differently to the prototype foamy-virus intasome. So the R1 sulfhydrobenzyl substituent actually bends back into a small cleft formed by the enzyme and makes intramolecular stacking interactions with the naphthyridine core. Likewise, if we look at the compound 4C when it's bound to the HIV intasome, the R1 pentanol group extends out toward the bulk solvent, similar to the compound 4F. However, when it's bound to the prototype foamy-virus intasome, the group again adapts a configuration that is shifted closer to the naphthyridine core. Here's a more direct comparison. On the left is a comparison of the binding mode of the compound 4F in the context of the HIV and the prototype foamy-virus intasome, and on the right is a comparison of the binding mode of the compound 4C in the context of the HIV and the prototype foamy-virus intasome. Now, in addition, we show that the binding mode of the more potent compound 4D it adapts a similar configuration as that of 4C, extending toward the bulk solvent. So what this is telling us is that if we want to understand how ligands bind, if we want to utilize the information for drug design or for understanding mechanisms of viral resistance, we have to be working with the correct construct. This is really critical. As I mentioned before, the residues in the active site are not necessarily conserved between prototype foamy-virus and HIV intasomes, and this is indicated in the table. Only about 40% of the residues are identical, and those are indicated in green. And many of these residues that are not conserved also happen to be viral resistant variants. And we know that drug resistance is a major problem. This is a table showing just a small subset of patient-derived viral resistant variants from INSTI usage in the clinic. There are many emergent drug-resistant variants and more that we are likely to see in the future. Here's just a look at some of the structural differences between the resistant variants. Each one of these panels shows an overlay of drug-resistant variants in HIV integrase and the corresponding residues of PFE integrase. And you can see that the conformations are not identical. The differences might be minor if you think in terms of atomic displacement. However, as I showed just a few slides ago, their effects can really be profound. So they can completely alter how the compound binds in the active site. So if we want to understand the atomic mechanism by which resistant variants displace the compound, again, we really need to be working with the real construct. So where are we going with this work? Well, many of you probably know that we can measure small molecule efficacy in cells using EC50 analysis. In the last several slides, I kept emphasizing the point about resistance. So if we look at how the naphthyridine compounds perform to inhibit HIV challenge in cellular assays, and particularly when the virus contains clinically relevant resistant variants, what one sees is that several of the naphthyridine compounds, especially compound 4D, outperform the clinically used drugs. Now, this is important because all of the compounds potently inhibit the wild-type virus. Even the poor compounds potently inhibit wild-type virus. However, what we're interested in is not necessarily inhibiting the wild-type virus. We've been doing that extremely well for the last decade. We're interested in how the compounds inhibit mutant virus that has already evolved resistance. That's what's going to be relevant for long-lasting therapy for patients. So we hope that the naphthyridine compounds may provide an avenue towards this goal. In the future, what we want to do is we want to understand how resistant variants affect INSTI binding. We want to decipher the atomic mechanisms that are involved in this process, and we want to use the cryo-EM pipeline for the purposes of drug design. So we believe that with structural biology, computational chemistry, medicinal chemistry, we can potentially make a dent in this area. I just want to finish up with this last slide. This slide describes the strategy that we are planning to take to achieve this goal. It's based on an idea called the substrate envelope hypothesis. This is an idea that was developed by Celia Schiffer's lab, and it was applied to the development of protease inhibitors about 15 or 20 years ago. The substrate envelope is the molecular envelope of an enzyme that binds natural substrates, so any and all natural substrates. In the case of protease, those substrates are the amino acids that are proteolytically cleaved by the enzyme. In the case of Integrase, natural substrates are both viral and target DNA. And these are displayed in the current figure. So a resistant variant needs to displace the bound drug. When a resistant mutation arises, if it displaces the drug while at the same time protruding into the natural substrate envelope, this can potentially compromise viral fitness. And therefore, the resistant variant has less of a chance to persist in the clonal viral population. So if you keep the drug constrained to the substrate envelope, then resistant mutants will compromise viral fitness and are less likely to be maintained in the next generation. Conversely, if the drug protrudes out of the substrate envelope, then it's easier for resistant variants to displace the compound while at the same time maintaining viral fitness. So therefore, as we're optimizing the compounds, we're now going to be using the HIV intasome substrate envelope to guide us and to make sure that we're constraining the molecule that we're modifying within its boundaries. To conclude, cryo-EM is experiencing an exponential growth curve. It's now applicable to drug design, although still relatively low throughput. We've shown that tilting the grid facilitates high-resolution structural studies of preferentially oriented samples. Intasomes are a very nice example of this. We've developed routine workflows for intasome inhibition and structural studies. And using these workflows, we can now get sub-3 angstrom resolution structures, which provide key insights into the structural differences, the modes of drug binding, and we're starting to investigate also the role of solvent and how that facilitates drug design. We've shown that using the correct specimen is absolutely essential. If you want to modify compounds and understand viral resistance, you really need to be using the correct specimen. And finally, the substrate envelope hypothesis, which is not a new idea, but is an idea that is now very relevant to improving drugs that are targeting HIV integration, especially in light of viral resistance. Finally, these are the people who did the work. Dario Passos and Ilona Jozwiak in my group have spearheaded the cryo-EM and the molecular modeling, respectively. This work is part of a long-standing collaboration with Min Lee and Bob Craigie at the NIH. We've since developed a strong team of collaborators in medicinal chemistry with the group of Terry Burke, virology with Steve Hughes, and computational chemistry with Stefano Forli. We're excited to be continuing all this work in the future with these folks. I'd like to thank the Scripps Research Institute, with whom we've established a consortium for utilizing the cryo-EM infrastructure, computational and cryo-EM support, both at Salk and TSRI, funding sources that have been generous to us over the last few years, And of course, Thermo Fisher Scientific, in collaboration with Nature Publishing Group, for giving me the opportunity to talk about this work. And last but not least, all the audience members who have signed up for this webinar. Thanks, and at this point, I'd be happy to take any questions.